All right. Angle. No, that way. Okay, so yeah, I want to start with this Punnett Problems 2 worksheet. Dealing with two characteristics when you're doing a Punnett square or talking about alleles and genes can be a little bit more confusing. So we are going to work through this together. I'll give you, if you haven't read through it yet, we're talking about dragons. They've been rediscovered, not really, but we are looking at the wing length and wing color of dragons. I thought this was more exciting than pea plants, so I went with dragons. The long wings are dominant to short, so that's the wing length. And green wings are dominant to gray, so that's going to be the color. So W is for the wing length, G is for the wing color. And we have a little bit of information and then have to answer some more information based on kind of what I described that you come across. So you have a male dragon and a female dragon. The male dragon is heterozygous for both wing length and wing color. So what's the genotype of the male? Who wants to tell me that? The male. Oh, okay. So male, what's the genotype of the male if he's heterozygous for both of those characteristics?
Is it both dominant traits? Big W and big G.
Those are there. There's also two more. So he's heterozygous for both of them. So that means, yeah, exactly. So big W, little w. So he's heterozygous for wing length and he's heterozygous for wing color. We did so much with just the single characteristic that it's easy to forget that when you have two characteristics, there's two alleles for each characteristic, so you have four, right? So it gets a little bit more complicated. So that's the male genotype. And then the next question, what are the possible alleles? So thinking about the alleles that the male can donate to his offspring. And we'll separate these by just commas. So what is one allele that he could donate? So what is one of these letters? What's that? Close. So those are two alleles. So he can, yep, exactly, yeah. So that's the confusing part sometimes. So one allele will be big W. He can donate a big W. He can also donate a little w. He can also donate a big G. He can also donate a little g. Those are all individual alleles. So this is something I didn't ask. Well, we won't go into it. Don't worry about it. So now the genotype of the female. She is, what did I say she was? She has short gray wings. So what is her genotype? Yep, exactly. So all littles, little w, little g, little g. So what would you call her? Instead of heterozygous, homozygous, recessive, yeah. So homozygous recessive is what you would call her. The male is heterozygous. Awesome, okay. So what alleles can she possibly donate? Just little w and little g, yep. So yeah, she can only donate two alleles. She only has two. She doesn't have the variety that the male has. Okay, so these are the alleles. And then these are the genotypes. Okay. So now we're going to cross them and see what the offspring might look like. Does everyone have this? I'm going to erase that. Good. Okay. All right. So we're going to put the father on top. So since there's a lot of squares potentially, right? So let's draw a big, big Punnett square here. And we'll put the dad, the male, on top. So what would, here we'll have, we have 16 potential squares, maybe. Okay, so this is our big Punnett square when we have two characteristics. So the male, his genotype was, we'll put his genotype up here. Big W, big G, little g, right? That's the male. We'll put him on top. The female is little w, little g, little g. So how do we set those up? Like what goes on one square? So if we're looking at the male, what would go here?
Big W, big G.
In that one square?
Oh, no.
Yeah, so one square.
This would be big W, I mean, big W.
Big W and big G. So he's going to donate both characteristics. So that's where it gets really confusing. So big, whoops, that's a weird G. Big W, big G will go on one. So to these potential offspring in this column, he can donate a big W and a big G, but he has to donate both of them. He can't just donate one of his characteristics. He has to pass on the characteristic for wing length, and he has to pass on the characteristic for wing color. So each of these squares has to potentially have one of each of those alleles from the dad. Does that make sense? That's where it gets confusing, trying to figure out which letters to put there. But each characteristic, you have to have one allele for each of those genes. So then what could we put here? So he has a lot of possibilities that he might be donating. Yep, little w, big G we'll put. So this is just going to be all possible combinations of his alleles, assuming you have both characteristics, right? So we have big W, big G, little w, big G. Okay, so we've got the big and the little w. So now we've got to do little g with the big W, right? And then we have to do little w, little g. So if you look at all of those letters, you have to combine both possibilities with both genes. Does that make sense? So you have to have one of each letter type. That's an easy way to think about it when you're putting together this Punnett square. You have to have a W and a G, or whatever letters you're talking about, on each square. You can't have two W's on one square, because then he's not donating one of the chromosome that has his G on it. So you have to have one of each characteristic when you're dealing with this two character or two gene situation. So always remember that. You're gonna have two different letters. So what about the female? Mm-hmm.
Would that be the same all the way down?
You don't have to do any more. Yeah. So that's the only option that she can donate. She can only donate a little w, little g. You could do it all the way down, but then it's going to be the same, basically all the way down, or all the way across. No, all the way down. Sorry. Yeah, so she can only donate a little w, she can only donate a little g. So when you combine those, then that's the only option for her gamete. So essentially what these are the gametes. So this will be her egg cell. That's all that can go into her egg. She doesn't have any other options of combinations between these. And then these are the dad's gametes. So we'll say, assuming dragons have eggs and sperm, those would be the sperm cells. So remember, when you have two characteristics, you have to have both characteristics accounted for on each of the lines or the columns, and just make all of the possible combinations. So when we listed out those different alleles that the female could donate, she could only donate a W and a G. The male could donate all of these, so we had to mix them and combine them in all possible ways. Okay, so now this is the easy part. The hard part really in these is setting up the parents. So setting up the or figuring out what the offspring are going to be, we'll have a big W, little w, and a big G, little g. So now for the offspring, we have to have 4 letters in here, right? Because when we combine 2 of them, if each has 2 letters, then we have to have 4 in the actual individual. So these are all possible individuals that we'll have.
Okay, so does it matter like if we put the W with the W or
No, it doesn't matter what order you put it in. I always put the big letter first, and then whatever, yeah, I mean, I put W first in both of these, so I put them first. It doesn't matter what order you put them in. So then we have little w, big G, little g, big W, little w, little, and then 2 little g's. Is everyone following me so far? Okay. This is the straightforward part, right? And then we have all littles. So little w, little g, little g. Those are the only 4 possibilities of offspring. We don't have all of the rest. So you can ignore all of this because the mom was homozygous recessive. It makes it a lot easier if you have an individual who's completely homozygous, whether it's dominant or recessive. It makes it a lot easier and you cut down on the number of squares. Okay, so now the last question was asking about the phenotypes. So I'm asking, what do these offspring actually look like with regard to their wing length and wing color? So let's just start on the left here. What would this individual look like? Would he have long wings or short wings, or he or she? Long, and then
green?
Green. Yeah, I was like, I don't remember what I mean dominant. Green. So yeah, long, green, right? So we have long green wing option. And then the next one, what's 2 little W's? What's that going to make it? Short wings. But what color? Green still, right? Because we have that big G. And I should have told you this is a typical complete dominant situation. It's not one of those complicated ones. So we have a long green, a short green, and then this third square over, what length wing is that? Long and gray. Yep, so long gray wings. And then finally, short gray, right? Yeah. Okay. And then did I ask, yeah, the phenotype ratio. Does anyone have a guess as to what that would be?
1.
Exactly. 1 to 1 to 1 to 1. Very easy. So there's 1 of each. So 1 of these to 1 of these to 1 of these to 1 of these. So 1 of each of the phenotypes. So make sure you have all of this information down, especially this cross, like the Punnett square, so you can look back at it and figure out how to set up the parents. Just remembering that you need 1 of each of the letters on each square, or each row or column, I should say. So you have to have a combination of the characteristics, but they can only donate 1 of their options at a time, like 1 W or 1 G. Any questions about that? Okay. I'll put the key on Canvas as well. You guys don't have to turn that in, so don't worry about that. All right, here's that one.
Study guide?
Oh yeah, thank you. I was actually going to say there is an exam coming up, and yeah, I haven't put that study guide up yet. I'll get it up today. Thank you for the reminder. That's weird. It's actually magnetic. Yeah, so we just have 2 more days of material. I might have to go a little bit into Friday with some of the new material. but because we're actually a day behind. So we'll see how far we get. Hopefully we can still do the exam on Monday, but we will see. Okay. Have that up. I actually cut a lot of material out of this linkage and mapping lecture. Oops, I need to share that with you guys. So I think I cut it a little further over the weekend. So if you have an older version, you may want to re-download it, but what's on Canvas is correct. Yeah, I put up the right one. All right. Yeah. So this is the very end of the genetics stuff. So we're going to talk about linkage and mapping. So what I just showed you, that hang on. All right. Can you guys see my screen? Hopefully, not you guys in the class, but for some reason, I can't see you guys. Wait. Hang on. I'm thinking I might have gotten cut off here. Sorry, guys. Okay, yeah. All right, I guess we're good. Yeah, so what I was just showing you was showing that basically those two genes weren't linked. The color of the wing and the length of the wing. It didn't really matter which one. You could basically mix them and match them. So today we're going to talk about when that doesn't happen, when genes are actually linked to each other. Just very briefly, and then how we kind of figure out if linkage is likely or not. Okay. So the law of independent assortment. This is one of Mendel's 2 laws that you need to know. This one deals with the fact that chromosomes are separating from each other during meiosis. So that means genes are separating from each other. So a gene on one chromosome, we'll say chromosome A, isn't necessarily linked to a gene on chromosome B, because they separate completely, supposedly randomly and independently. So like I said, in theory here, genes on different chromosomes aren't linked to each other. So just because, I think I've used this example before, just because you have blue eyes doesn't mean you have to have blonde hair. So that would be an example of non-linkage. They can mix and match. So we look at this kind of basically the Punnett square we just did, but with P, color and texture or shape instead of the dragon wings. So this is kind of the same setup. We have a parent who's homozygous recessive and then a dihybrid parent, so a parent who's heterozygous for both characteristics. What you have, and I didn't talk about this on the previous Punnett square, but you have 2 individuals that have the exact same phenotype as the parents. So those are called parental type offspring. So this big Y, little r is exactly like this parent. Big Y, little r, big R, little r, yeah. So exactly like one parent, and then this P individual is exactly like the other parent, little y, little r, little r. little y, little y, little r, little r. So they're the exact same genotype. So that makes them what we call parental type offspring. Same genotype as the parent, one or the other. Where you get that genetic diversity is where you have the recombination of these alleles. So here we have a big Y, little r, which is not like either of the parents, right? Their alleles kind of mixed with each other. So we no longer have a smooth yellow pea, we have a wrinkled yellow pea. And then the same for this square over here. This one is also not like either parent. So those are called recombinant offspring. The alleles are recombined, recombinant. Okay, so recombinant versus parental type. We're going to look at the ratio of those two to figure out eventually if there's some kind of linkage between characteristics or not, between genes. But here it's a 50-50. So likely no linkage between the shape of the pea and the color of the pea. Oh, yeah, sorry. I have additional notations here. So you can go back and know what the heck I was talking about. So recombinant offspring, new combination of traits. The phenotype is not like either parent. It's a combination, a mixture of the two parents. Here we have to assume we're setting this Punnett square up as if Y and R are not linked, that they assort independently, because we have all possible combinations, right? We're not assuming that Y and R are linked in any way. That's what we've been doing this whole time, just assuming that they're not linked. This shows, like I said, a 50-50. Half of the individuals are parental type and half of them are recombinant offspring. So that would be a 50% recombination frequency. So we'll look at those percentages between the recombinant and the parental type to figure out if there's linkage. Okay, so we need to go back to crossing over very briefly. Remember, crossing over is where the homologous chromosomes, so the father's and the mother's chromosomes that have the same genes on them, match up, and then they switch sections, right? They'll like cross their little arms or legs, and they actually switch pieces of their chromosomes with each other. So it mixes everything up. So if you look at these two sets of chromosomes, we have the A gene way up here and the B gene way down here, versus here we have them really close together. The A and the B genes are very close together on these chromosomes. Which of these scenarios, A or B, so far apart or close together, will crossing over be more likely to actually happen in between the two genes?
B.
Which?
B.
Not B, A. So if you think about the chromosomes switching, or the, sorry, sections of the chromosomes switching, or maybe this will help. There's a lot more area in between A and B for that to happen. So it's more likely that they'll actually switch and be mixed up versus here. There's not really a lot of area. You'd have to have crossing over happen right here, a very specific spot. So it's a lot less likely to happen just based on chance. So you have all of this area in between these two genes on this chromosome that would allow for crossing over to happen anywhere in here. So the chances are a lot greater that you have that recombination occurring. So for you guys on Zoom, if you couldn't see that, you have a lot more area between the two genes. If they're further apart on a chromosome, they're more likely to be mixed up and recombined. When you just have a tiny bit of space in between the chromosomes, the crossing over has to happen, like I said, in a very small area. which just by chance is less likely to happen. There's just not as much area for that crossing over to happen to actually recombine those alleles. Does that make sense? More area means more likelihood of crossing over. So this is the take-home message from that. The farther apart genes are on a chromosome, the more likely the chance of recombination. And here we're assuming, and this is usually the case, that crossing over, where that happens is totally random. Okay, so further apart, if the genes are further apart, you have a greater likelihood of recombination through crossing over. Now getting into linked genes, basically genes that are pretty close to each other. This is what we're talking about. Genes that are really close to each other, it's unlikely that they'll be mixed up due to crossing over. They're gonna most likely stay with each other and not be recombined. So linked genes are just genes on the same chromosome that are usually inherited together. So in other words, they're close to each other. They don't get mixed up a whole lot. And these don't follow the typical inheritance patterns that we've been talking about. Because they're linked, it's not like you don't have all possible combinations, like I was just saying, of the allele combinations that we were doing with the dragon example. You don't have all of those as a possibility, because if those two genes are linked, the W and the G genes that we were talking about, the wing length and the wing color, if those are linked, they're likely not going to mix up where it's an equal possibility of getting a dominant or recessive of each combination together. So a general rule of thumb is if you look at the number of recombinant offspring or the frequency, if less than half of them, so less than 50% are recombinant, linkage is likely. So you would expect, like I showed you on that first slide with the pea plants, totally randomly with no linkage, half of the offspring would be parental type and half would be the new combination, the recombinant offspring. If you get less recombination, you probably have some kind of linkage going on where one gene is sort of almost attached to another gene, so they both get inherited together. So that's all linked genes are. They're close together, they tend to be inherited together. I really did cut out a lot. So you can actually figure out how far apart genes are based on recombination frequency. That's kind of the information I cut out of this. So we're not actually going to look at how to map genes on a chromosome. But these are kind of those are the main take-home messages that I want you to know about linkage between genes. The next part of this is just about essentially mutations that can happen. And I cut some of this information out too, but I want to talk about some of the major categories of mutations because this doesn't always happen perfectly. Meiosis doesn't happen perfectly. I already talked about aneuploidy, which I'll talk about here at the beginning again, where you have an incorrect number of chromosomes in a gamete, etc. So that'll be the rest of this, and then we'll get into the next chapter, which is DNA replication. So these are all the types of chromosome mutations. I'll just go through them kind of one by one. So here, I'm mostly talking about large-scale mutations, such as aneuploidy, which I defined before. It's just where there's an abnormal number of chromosomes. So too many or too few. So you can have an extra chromosome or multiple extra chromosomes or too few. Doesn't really matter what direction it is. It's just an incorrect number. This happens, as I said before, when there's non-disjunction. So during meiosis, you have all of these separations, right? So you have the homologous chromosomes together, they'll separate, and then you have the sister chromatids in meiosis 2 separating. So at some point, one of those separations doesn't happen correctly, and the number of chromosomes or chromatids isn't split in half evenly into each eventual cell. They kind of all go to 1. So you don't have the right number when you get to the end of meiosis. So yeah, I don't really need to go into any more detail on that, but here we have too many chromosomes where you should just have 2. This is normal. 1 too many chromosomes, which then throws off the final, rather than having 4 chromosomes, you have 5. Down syndrome is probably the most commonly or well-known type of aneuploidy. And it's also called trisomy 21. Does anyone want to tell me based on, so these are all the chromosomes of a person with Down syndrome. And you can kind of see them numbered 1 through 22, and then the sex chromosomes here, X and Y. Why is it called trisomy 21? You can figure it out based on the chromosomes. Exactly, yeah. The 21 place, yes. So the chromosome 21, there's 3 here rather than 2. So remember, each of these sets of chromosomes, you have one of them comes from your mom, one of them comes from your dad. So you should only have 2. Here at chromosome number 21, for some reason, non-disjunction occurred. And it occurs at chromosome 21 in Down syndrome very well every time, obviously, if the individual has Down syndrome. So it's a common location of non-disjunction that shows up in many people. I mean, it's not a common syndrome, but it shows up always at chromosome 21. Non-disjunction occurs, so there's 3 chromosomes instead of just 2. So you get 2 from one parent, 1 from another parent. And that's what causes Down syndrome, which I think most people are generally pretty familiar with. So trisomy 21, the tri means 3, obviously. Trisomy 21 at space 21, chromosome 21. So that's the most common non-disjunction, or the most well-known probably, result of non-disjunction. You can also have inversions and translocations, which basically means genes on a chromosome are in the incorrect place or incorrect order. So inversion is at the top here. If we look at this chromosome, we're going to imagine that it has genes A through H on it. Normally it goes B, C, D right here. In an inversion, those are just flipped. So you get D, C, B. So they're in the opposite order. They're all still there. They're on the right chromosome, but the order is flipped. And usually inversions don't cause many issues because you have all the genes there. They're still coding for the proteins. They're doing what they need to do. They're just in a different order. So sometimes they can cause issues, but no major issues typically are caused by inversions. Translocations cause bigger issues. So a translocation is down here where you actually have genes moving. So a segment of a chromosome moving from one chromosome to another, but not a homologous chromosome. So to another chromosome that has a totally separate set of genes. So here you can see you have on this chromosome here, AB. On this chromosome, you have MNO. Those are going to switch. Those sets of genes will switch. They code for different proteins. They do totally different things. So if they separate, and they should, during meiosis, you're basically missing some of your genes that you should have, right? So translocations can cause some pretty major issues. As I noted here, there's a chronic form of leukemia, which is blood cancer, that's caused by a translocation between chromosomes 9 and 22. That doesn't really mean a whole lot to you, but just this translocation of genes between these two chromosomes causes a pretty dangerous condition. Right, and then we have duplications and deletions. Duplications are just where a segment of a chromosome is completely copied. So you have two of those in one chromosome. Can cause some issues. A deletion is where a segment of a chromosome is totally removed and just gone. It doesn't go to another chromosome, it's just deleted. You can imagine, depending on what segment of a chromosome gets deleted, that can be a pretty significant issue. You're missing those genes that were there, which means whatever proteins those genes were coding are no longer able to be created or coded for. So deletions are really, really dangerous. There is a specific deletion that I have here. You don't have to know the name. They name genes and segments of chromosomes based on a kind of complicated method. So 22q11.2 deletion. So a very specific part of a chromosome that gets taken out, and you can look at how they name them if you want to, but you don't have to worry too much about it. But a segment has been taken out. So this is a normal chromosome, and that whole blue section was essentially deleted. So whatever genes were in there are gone. So it can cause a lot of different effects, as you can see here. Just this one deletion: heart defects, cleft palate, problems with the immune system, developmental delays, seizures, learning disabilities, autism spectrum type behaviors. So just with that one deletion, you can possibly have all of these or a combination, a subset of some of them. So deletions are usually not a very good thing. All right, I think that wraps this up. Yes. All right. So we're just going to quickly get into the next chapter. That's what I should have finished on Friday or Wednesday. The last chapter we're talking about is DNA structure and replication. So we're getting back into some kind of, I guess it's still been molecular, but smaller molecular type stuff. This is probably the harder part of this unit, I would say. So we're going to learn how DNA actually duplicates itself and makes more of itself. All right. All I'm really going to have time to talk about today is the structure aspect. So we'll kind of review DNA structure. Hopefully a lot of this looks kind of familiar because we talked about it when we talked about the biological macromolecules, the nucleic acids. So DNA stands for deoxyribonucleic acid. I think everyone knows this is how we inherit characteristics from our parents. It's essentially what codes for everything in us, all of the proteins that make us function. And as a reminder, I think I went over this as well briefly. We're going to talk about this in detail now, the central dogma of biology. So this is how a essentially proteins are made and how we inherit all of this information. So DNA is going to code for RNA. That's just ribonucleic acid. We'll talk about that plenty. So deoxyribonucleic acid, ribonucleic acid, and then RNA will make proteins or code for proteins. So this is all about coding. So you want to memorize that order, DNA to RNA to protein. We start with DNA, then we make RNA, and then we make proteins. We talked about DNA replication when we talked about the cell cycle. So this has to happen for every single cell that is created during mitosis and meiosis. We have to replicate the DNA first in order to create new cells. Otherwise, one cell just doesn't have any DNA in it. If you're making two cells from one, you have to have DNA being replicated. So that's what we're talking about here for the rest of this unit, hopefully finishing on Wednesday. And as a reminder, the monomer that makes up DNA, so DNA is this really long strand, right? Those are made up of, it's made up of individual nucleotides. The nucleotide has a few different parts to it. The phosphate group, the sugar. In DNA's case, it's deoxyribose. That's the name of the sugar. It's always deoxyribose. So you have phosphate group, sugar, and then the really important part that we're going to focus on, the nitrogenous base. And that's why I bolded it. So the nitrogenous base is sort of that's where the code comes from. Every single nucleotide has a phosphate and a deoxyribose. The only thing that differs is the nitrogenous base. And there's 4 nitrogenous bases in DNA. This is our code. The A, the G, the T, the C. Adenine, guanine, thymine, and cytosine. So there's only 4 letters that create this code that essentially make you. It's pretty crazy. So you want to know those names, and obviously their abbreviations are just the first letter, so it's pretty easy to remember. But I'll be referring to them by their name as well as just their letter abbreviation. You don't need to know, I'll go into a little bit more detail about some of them on the next slide, but you don't need to know a lot about them other than the few things that I tell you here. Okay. Oh, sorry.
Oh, I got it right.
You got it? Okay. Yeah, cytosine. So if we're looking at a strand of DNA, here we have, so this is 1/2. I'll talk about the double helix aspect, but this will be one strand of a bunch of monomers stuck together, right? So this is a lot of nucleotides, well, not a lot, 4 nucleotides bonded together. So you have one nucleotide here, one nucleotide here, etc. So you have the nitrogenous base, the sugar, the phosphate group with each of them, and they all get strung together. This is where we get into naming the ends of the strands, which seems crazy and unnecessary, but it'll come into play when we get into how DNA actually grows and replicates itself. So for now, really all you need to know is that there's a 5' end and a 3' end. So this little apostrophe just stands for prime. This is how chemists name the molecules, basically. The 5' end has a phosphate group coming off of it. So if we're talking about a string of nucleotides strung together, the 5' end has the phosphate group. The 3' end of that strand has an OH, which you can't really see, but it's right here off of the phosphate. So you have the OH on this side. as this strand grows, new nucleotides are added to the 3' end. That's the really important part. New nucleotides, if you're growing a DNA strand, you're building a new strand, are added to the 3' end. I probably won't ask you if the phosphate or the OH end is the 3' or 5'. You want to know that a DNA strand grows in the 3' direction, which just means new nucleotides are added to the 3' end. So this would be growing down. No new nucleotides can be added up there. Oh yeah. And the bond, we have a couple different bonds in DNA. Between the nucleotides on one strand, it's a phosphodiester bond. That's the name of it. So here where you have this nucleotide bonding to this next one, that's a phosphodiester bond. The other bond will be a hydrogen bond, and I'll talk about that. I'll get to it till Wednesday. Or maybe I'll get to it today. So the secondary structure, let's stop there. We'll talk about the secondary structure, which is basically the double helix on Wednesday, and hopefully get most of this finished up. I'll get the study guide up today. And yeah, I'll see you guys on Wednesday.